Thank you very much for the presentation. Good morning once more, dear ladies and gentlemen. My talk is devoted to the discussion of the ligand affinity and amount on the biological performance of radiopharmaceuticals, and here it's exemplified with gallium-dotatok and gallium-dotatate. And the essence of this talk is to stress one more time the complexity of the translation of the observations from in vitro to in vivo conditions. Receptor targeting peptide-based tracers of chelator-mediated radiolabeling are usually consist of at least three principal components. And it is the vector molecule for the binding to specific binding to the target, chelator moiety for the binding of a radionuclide and radionuclide for sending the signal. And concentration of positron-emitting radionuclides is usually minute. And thus the excess of the ligand is usually used to accomplish the radiolabeling chemistry. And that results in a very small fraction of the total amount of the ligand actually carrying the radionuclide. And the common perception is that high affinity peptide-based tracers should be labeled and administered with high specific radioactivity. And that is a very viable requirement because of the large excess of non-labeled counterparts can easily outplay the labeled tracer. And however, both preclinical and clinical studies have demonstrated very complex translation from in vitro to in vivo, and even translation between the culture cells and tissues, and also between the different species. So binding affinity is usually determined in cell cultures in vitro, which means that the physiological factors are excluded. And in this particular case, we have two analogs with seemingly minor difference. And we have threonine amino acid residue where dotatate has carboxylic group, and here in dotatok we have hydroxyl group. And that difference resulted in 10-fold difference in IC50 value as determined in transculated cells expressing somatostatin type 2 receptors. However, the determination of IC50 value of octreotide against these two tracers in tissue of frozen section radiography of rhesus monkey brain that expresses somatostatin receptors did not show any statistically significant difference. And the experiment was also conducted ex vivo in biodistribution in rats, looking at such organs as adrenal, pituitary, and pancreas that express physiologically somatostatin receptors. And again, no statistically significant difference could be found in terms of radioactivity uptake. And additional experiment was conducted in order to investigate the influence of the amount of the injected peptide. And here again, the degree of the drop of the uptake and also the pattern was similar for two tracers. The comparative study of gallium-dotatate and gallium-dotatok was also conducted clinically, wherein each patient was examined 2 times, 1 week apart, administered once with dotatok and second time with dotatate, or vice versa for randomization of the study. And no statistically significant difference could be found in terms of detection rate and uptake degree. So in occasional patients, the singular liver metastasis could be detected by DOTATOC and not by DOTATATE, and vice versa. In some cases, it was detected by DOTATATE and not by DOTATOC. And these graphs present SUV values and where each patient is presented with the lesion of highest uptake. And here we have the DOTATOC in red and DOTATATE in blue at 1 hour, 2 hour, and 3 hour post-ejection as measured at whole body examination. And again, no statistically significant difference could be found between these two tracers. So these graphs present the radioactivity accumulation as a function of time in tumor, liver, spleen, kidney, as well as clearance from the blood. And these values are the mean values from all the patients presented again by the highest, by the lesion with highest uptake. And blood clearance was fast and almost identical for the two tracers. And plateau was achieved just after 50 minutes. And these graphs are very interesting in terms that they present the tumor to liver, tumor to spleen, and tumor to kidney ratio. And no statistically significant difference could be found for the ratio of tumor to spleen, but statistically significant difference could be found at time point 3 for tumor to liver ratio and at all time points for tumor to kidney ratio, and it was higher for DOTATATE. So the dosimetry study did not show any statistically significant difference with regard to total effective dose, and it was basically identical for the tracers, but it was slightly higher absorbed dose for the kidneys and liver for DOTATATE, slight but still statistically significant. So from this affinity comparative study, we can conclude that no statistically significant difference in tumor uptake and detection rate could be found for the two tracers. So it means that both tracers can be used equally well for staging and patient selection for subsequent therapy. However, higher absorbed dose to kidneys with DOTATATE may render gallium DOTATATE more relevant with regard to the radiotherapy planning using lutetium DOTATATE. The influence of specific radioactivity on the binding of gallium DOTATOC was investigated in vitro, again in frozen sections of rhesus monkey brain expressing somatostatin receptors, and these graphs present the scattered plots for the determination of KD and Bmax values. And you can see that in this region, specific binding is considerably higher than non-specific binding, and this region would be most appropriate for the accurate quantification and would provide the highest contrast. And on the contrary, this region would be, and specific radioactivities corresponding to this region would be inappropriate and would not, due to the poor contrast and consequently not impossibility to do the accurate quantification. So to make it more illustrative, the same data is presented here in a bit different way. Here we have the ratio of receptor-bound tracer over the free concentration of the tracer as a function of specific radioactivity. And this ratio also presents the signal to background ratio reflecting the contrast of an image. And we can see that the contrast of an image would be deteriorated when specific radioactivity is approaching zero. And this dependence is very critical around the inflection point where the slight difference in specific radioactivity may bring up a much more drastic difference in contrast of the image. At certain value of specific radioactivity, this ratio becomes independent on the variation of specific radioactivity, and this region would provide higher reproducibility and robustness of the biological assays, because the variation of specific radioactivity from one experiment to another would not influence the accuracy of the quantification. And please note that this value is 100 megabecquerel per nanomole. However, when we look at the in vivo distribution, in this case in rats, we see that the decrease in specific radioactivity from 50 to 2 megabecquerel also brings the blocking effect and decrease in uptake 4 times. However, a very good contrast and accurate quantification could be possible already at 15 megabecquerel per nanomole. And further on, the influence of specific radioactivity was also on the biodistribution, was also studied clinically with the intention to estimate the receptor density in vivo and also to optimize the imaging quality. And in this case, these are 3 sequential examinations conducted with gradually increasing amount of the injected peptide and thus decreasing specific radioactivity, conducted in the same patient on the same day. And you can see that the increased amount of the total injected peptide increased radioactivity uptake in the lesion. At the same time, it decreased the uptake in liver and spleen, and further increase brought up the blocking effect. So, as we can see from this experiment, it was possible to find a more or less optimal condition when we could have the higher uptake in the lesion at the same time to reduce the radiation burden to healthy organs. And interestingly, the specific radioactivity corresponded to only 2 megabecquerel per nanomole, and the highest specific radioactivity was not the best one. So, but I will go just back. So, but it's not actually that simple and straightforward, and for all patients except for one, this uptake pattern was the same, but for one patient, the uptake increased even with the highest dose of the peptide, and this is a very striking example that stresses that the patient treatment should be individualized, because in this case, most probably the therapeutical planning would be very different from the other patients. So in conclusion, difference in affinity determined in cell cultures might not be manifested in vivo in terms of radiopharmaceutical biodistribution, targeting, and imaging properties. Lower specific radioactivity might be more advantageous in vivo in terms of target uptake, image contrast, and healthy organ radiation dose. The preclinical and clinical studies demonstrated necessity for the optimization of the specific radioactivity of the radiopharmaceuticals for the personalized patient management. Thank you very much for your attention.
Thank you very much, Irina. This paper is open for questions. Rich Wall.
The data are very interesting. Could you comment on the mechanism that you think might be in place for the reduction in hepatic activity? Have you looked at receptor binding or nonspecific binding to liver? In other words, why do you think the liver effect is occurring? And then secondly, does order make a difference? That is, if you gave the same mass first, followed by radioactivity, would the effect be the same as if you combine a low specific activity preparation? So simultaneous as opposed to sequential dosing. So 2 questions.
Yeah. The second question, that would depend on the time between the two injections, and if you are sufficiently close, it will be most probably the same. I would believe that it would be the same effect. The first question, it's actually quite tough one, because the same phenomena has been also demonstrated or observed in antibody field, and also with other peptide-based tracers that we have been using. And the thing is that I don't think there are any studies available on the mechanistic investigation. And it's only the hypothesis that we have so far. And it could be that when we have the healthy tissue and you have the physiological expression and of the receptors in healthy tissue, then most probably the kinetics of the tracer binding to the receptors expressed on healthy tissue and on the tumors is different. And it can be also maybe reduced to the hypothesis that when you have the healthy tissue, you have only the diffusion, and when you have the tumor tissue, you have both diffusion and receptor binding. So basically what happens, the tracer goes into healthy tissue and it comes out. And then it is available for but in the tumor cells, it goes in and it doesn't leave the cells. So, and basically, I think this is also can be observed when people do several time point imaging, and they see increased uptake in the tumors and decreased uptake in the healthy tissue, which means that when the receptor is liberated from the healthy tissue, it's still available for the tracer to take it up. So yeah, those are only the hypotheses. So far, I don't think any studies has been performed.
Do you think peptidases might play a role in some of this as well?
So you mean the tracers are getting metabolized, and it's the catabolites that enter the cells? And what did you mean with that? You mean the uptake in the tracers, or you mean the uptake in the healthy tissue?
Well, if the peptides are being snipped up.
Actually, it has been shown that somatostatin analogs that are usually used, DOTA-NOC, DOTA-TATE, DOTA-TOC, are quite stable in vivo. So basically, within the time frame that we have with gallium-68, and it's up to 4 hours, those tracers are stable.
Okay.
So those are not catabolites. But I would also suspect that liver is also playing a role as a filtration organ in them. So maybe as an answer to the previous question, that might also play a crucial role in the uptake in liver.